Hey guys, how's it going? This is FixerMed. Welcome back to my high yield genetics review series for the USMLE Step 1 NBME CBSE and NBME CAS examinations. This will be part 7 of my multi-part video series covering the entire discipline of genetics from start to finish in a comprehensive but high yield manner. So without further ado, let's go ahead and get started on today's content. The main focus of today's lesson is going to be about population genetics. Population genetics is a study of genetic variation and evolutionary processes within populations. Understanding population genetics helps explain how genetic traits are inherited, maintained, and changed over time in populations. Some key concepts to remember about population genetics include genetic equilibrium, genetic drift, founder effect, and population bottleneck. Moving on. First off, let's go ahead and discuss the Hardy-Weinberg Equilibrium. The Hardy-Weinberg Equilibrium describes the theoretical state in which allele and genotype frequencies remain constant from generation to generation in an idealized, infinitely large, and randomly mating population. The main equation for Hardy-Weinberg is p squared plus 2pq plus q squared equals 1, where p and q represent the frequencies of the two alleles in the population. Some assumptions you had to make with Hardy-Weinberg equilibrium include the fact that there's no mutation, no migration, no natural selection, random mating, and infinitely large population size. Some applications of Hardy-Weinberg equilibrium include the fact that it provides a baseline for comparing observed allele and genotype frequencies in real populations, which helps identify factors such as selection, migration, or genetic drift affecting the population. Moving on, let's talk about genetic drift. Genetic drift refers to random fluctuations in allele frequencies in a population due to chance events, particularly in small populations. The primary mechanism of genetic drift occurs due to sampling error during reproduction, which leads to the loss of genetic diversity over time. The main effects of genetic drift is more pronounced in small populations and can lead to the fixation of alleles, which means the loss of genetic variations, or the loss of alleles, random genetic drift. Some consequences of genetic drift include the fact that it can increase genetic differentiation between populations and increase the risk of inbreeding depression or loss of adaptive alleles. Moving on. The founder effect. The founder effect occurs when a small group of individuals migrate and establish a new population, resulting in a loss of genetic diversity compared to the original population. The main mechanism of the founder effect results from a limited number of founding individuals carrying only a subset of the genetic variation present in the original population. Some examples of the founder effect include the Amish community in the United States and the Afrikaner population in South Africa, both exhibiting increased frequencies of certain genetic disorders due to founder effects. Some consequences of the founder effect can lead to increased frequencies of rare alleles and genetic disorders in founder populations, affecting their genetic diversity and evolutionary potential. Moving on. Next, we have the population bottleneck. A population bottleneck occurs when a significant reduction in population size reduces genetic diversity due to a sudden event, such as natural disasters, disease outbreaks, or human activities. The main mechanism of the population bottleneck is when bottlenecks reduce the number of individuals contributing to the gene pool, resulting in a loss of alleles and increased genetic drift. Main effects of population bottleneck can lead to a loss of genetic diversity, increased levels of inbreeding, and decreased adaptive potential in affected populations. Some examples of population bottleneck include the northern elephant seal and cheetah populations, which experience severe bottlenecks, resulting in reduced genetic diversity and potential health issues. Some conservation implications of population bottleneck include the fact that you have to understand and mitigate the effects of population bottlenecks for conservation efforts that are aimed at preserving genetic diversity and population viability. Let's go ahead and move on. So we have a NBME style question here, In a small isolated population of red foxes, a genetic study reveals a significant decrease in genetic diversity over several generations. Which of the following evolutionary mechanisms is most likely responsible for this observed reduction in genetic diversity? All right, I think I gave you guys enough time here. If you need more time, please feel free to pause the video. Otherwise, moving on now. A population bottleneck occurs when a significant reduction in population size reduces genetic diversity due to a sudden event such as natural disasters or disease outbreaks. In this scenario, the observed decrease in genetic diversity in the small isolated population of red foxes suggests a population bottleneck as the likely mechanism. Population bottlenecks lead to a loss of alleles and increased genetic drift, resulting in decreased genetic diversity over time. Therefore, the correct answer is D, population bottleneck. Moving on to see why the other answer choices are wrong. Hardy-Weinberg Equilibrium describes a theoretical state in which allele and genotype frequencies remain constant in an idealized, infinitely large and randomly mating population. It does not account for changes in genetic diversity due to population size reductions. Genetic drift refers to random fluctuations in allele frequencies in a population due to chance events, particularly in small populations. While genetic drift can lead to a decrease in genetic diversity, it is not specific to sudden population size reductions. The founder effect occurs when a small group of individuals migrates and establishes a new population, resulting in a loss of genetic diversity compared to the original population. However, in this scenario, there is no indication of a founder population establishing a new population. Gene flow refers to the transfer of genetic material from one population to another through migration or interbreeding, which can increase genetic diversity. However, in the context of a small isolated population experiencing a decrease in genetic diversity, gene flow is less likely to be responsible for the observed reduction. So, hope that helps you guys out. I think gene flow isn't really high yield, but seeing the wrong answer choice here might help you in a future question, even though it might be low yield information. Moving on. In a small population of deer on an isolated island, a group of individuals migrate from the mainland and establishes a new population. Over time, the genetic composition of the island population becomes different from that of the mainland population, with certain alleles being more common and others being absent. Which of the following evolutionary mechanisms best explains these observed differences in allele frequencies? Pretty straightforward since we just went through the wrong answer choices, so I actually think I'm going to move on now. If you need more time, please feel free to pause the video. Moving on now. The founder effect occurs when a small group of individuals migrate and establish a new population, resulting in a loss of genetic diversity compared to the original population. In this scenario, the observed differences in allele frequencies between the island and mainland populations following the migration event suggest the founder effect is the likely mechanism. The founding individuals may carry only a subset of the genetic variation present in the original population, leading to differences in allele frequencies between the two populations over time. Therefore, the correct answer is C, founder effect. Let's see why the wrong answers are wrong. Hardy-Weinberg Equilibrium describes a theoretical state in which allele and genotype frequencies remain constant in an idealized, infinitely large, and randomly mating population. It does not specifically address the establishment of new populations or changes in allele frequencies due to migration events. Genetic drift refers to random fluctuations in allele frequencies in the population due to chance events, particularly in small populations. While genetic drift can lead to changes in allele frequencies, it does not specifically describe the establishment of new populations or the loss of genetic diversity following migration events. Population bottlenecks occur when a significant reduction in population size reduces genetic diversity due to sudden events such as natural disasters or habitat destruction. However, in this scenario, there is no indication of a sudden reduction in population size following the migration event. Gene flow refers to the transfer of genetic material from one population to another through migration or interbreeding, which can increase genetic diversity. In this scenario, gene flow from the mainland to the island population may contribute to changes in allele frequencies, but the establishment of a new population and subsequent loss of genetic diversity are better explained by the founder effect. So that will do it all for today's video. If you have any questions, comments, or concerns, please feel free to leave them in the comment section below. I would also appreciate it if you guys could leave a like and potentially subscribe to the channel. It really helps out a lot. If not, this is FixerMed signing off. Be sure to have a great day, everyone. Good luck studying, and goodbye.